The CGEN's 2023 cohort class at La Sierra University conducted annotations and characterizations of genes within Microbacteriophage Jeeves. Jeeves is a separate phage assigned to cluster 814 that infects Microbacterium smegmatis hosts. Our cohort worked to investigate and analyze the complexities of phage Jeeves. Initially, we generated a substantial number of Jeeves recombinant plasmids via molecular cloning, followed by a sequence of phenotypic assays. Molecular cloning was necessary to create a stock of recombinant plasmid backbones containing gene targets of interest. The p-extra plasmid was used as it could be integrated into E. coli and M. smegmatis genomes, which allowed us to do both molecular cloning and cytotoxicity assays. Additionally, the p-extra plasmids harbor an inducible TEP promoter upstream of the phage gene sequence. The promoter becomes activated in the presence of anhydrotetracycline inducer, or ATC, which allows us to dictate when our target genes are being expressed. These recombinant plasmids were used to perform phenotypic assays which identified the observable traits and behaviors of each gene in response to various changes in stimuli. One type of phenotypic assay pivotal to our research was the cytotoxicity assay, a technique used to assess the impact of phage gene expression on the host cell growth. In these assays, gene products leading to a reduction in bacterial colony size are classified as toxic, whereas those seen to have no reduction in colonies were classified as non-toxic. By conducting cytotoxicity assays on all genes numbered 1 through 52, excluding gene 7 within phage Jeeves, and varying the concentration of ATC across 3 levels, we were able to successfully identify gene products that exert toxic effects on M. smegmatis. This comprehensive analysis not only enhances our understanding of the genetic makeup of phage Jeeves in the context of M. smegmatis, but it also holds significant implications for future research and medical endeavors.
Our cohort isolated 52 genes from phage Jeeves and inserted them into Mycobacterium smegmatis to perform cytotoxicity assays. Molecular cloning was first performed to create a stock of plasmids containing target gene inserts for the subsequent phenotypic assays. In order to do this, each gene was first amplified via PCR from phage lysate and checked through gel electrophoresis. The DNA was then purified and assembled into p-extra plasmids. The recombinant plasmids were transformed into Escherichia coli, colony PCR was performed, and successful colonies were incubated. The plasmids were then extracted from the E. coli and transformed into M. smegmatis via electroporation. These colonies were used to make serodilutions, which were then used to make cytotoxicity assays for observation. Toxicity was determined by comparing the amount and size of colonies present on the plates with and without ATC, which is an inducer for the genes of interest.
Genes 1 through 52 were successfully isolated from bacteriophage genes and cloned into recombinant plasmids. Cytotoxicity assays were successfully performed for 49 of the 51 cloned genes using these recombinant plasmids. Genes 2 and 36 failed to produce colonies following electroporation, and thus the toxicity of these genes could not be determined. On the left, we see that of the 49 genes that were characterized through cytotoxicity assays, 31 were determined to be toxic when expressed in M. smegmatis hosts. This finding was significant to us, as we didn't expect over half of our genes to be toxic. For reference, only about a third of the phage genes examined by previous cohorts at our institution were shown to be toxic. We found that not all toxic genes were built quite the same, as represented by the figure in the middle. 15 of these toxic genes resulted in a mild reduction of colony growth, with half of these mildly toxic genes having no known function. The majority of the mildly toxic genes were involved in the structural assembly of the phage tailor head. 8 of the 31 toxic genes resulted in a reduction of colony growth between 1 to 3 logs, and included various genes that were related to different areas of the phage's structural assembly. The last 8 toxic genes were determined to be very toxic to the M. smegmatis host, resulting in colony growth reduction of greater than 3 logs. These genes were mostly involved in tail assembly or had no assigned function. As shown in the figure on the right, the majority of toxic genes either played a structural role in phage assembly or had not yet been assigned a function. Although genes that did not have a function were better characterized through the study, specific functions could not be assigned.
A vast majority of genes found within bacteriophages remain uncharacterized, hiding a plethora of novel functions applicable to the many fields of science. In the case of the toxic characterization of genes in Jeeves, a peculiar characteristic stood out, which is expanded on later. Although assigning toxicity levels to most of Jeeves' genes was possible, genes 2 and 36 remain unclassified. By analyzing alternate experiments, we may be able to identify the purpose of these genes. In fact, all of the genes may benefit from additional testing to help determine their genuine function. One such test we can run is the defense assay. Through this assay, a gene's protective function can be evaluated. Protecting the host bacteria is vital for any phage, as it prevents competition with other phage particles. Using this procedure, we may determine which genes are capable of this process from within phage Jeeves. Another test to further characterize both known and unknown genes would be to perform interaction assays. It should be noted that cytotoxicity assays involve the expression of a singular gene product within its host. Interaction assays would allow for the study of protein-to-protein interactions of said gene products to observe more accurate interactions between proteins that one might see in nature rather than in a controlled lab environment. Lastly, comparing the number of toxic genes from different phages within the same families as Jeeves may provide an explanation as to why such a large majority of genes were toxic. Phage Jeeves in particular exhibited an unusual characteristic. A large portion of the genes were found to be toxic when compared to phages such as Amelie, Hami, and Gur from clusters K1, K6, and F1 respectively. Phages belonging to the same cluster as Jeeves, A14, are still undergoing characterization and may reveal a pattern unique to the A14 cluster. Overall, this research leads to new and exciting avenues within the realm of phage biology.
Thank you.